Oh, good morning, everybody, both here in the classroom and also people tuning into the webinar. Today is lecture 3 of the drug discovery program for the pharmacology course here. I'm going to be talking about the biologic side of things. I know for the last couple of lectures you've been listening to the small molecule aspects, but I'm going to talk mostly about biologics, chiefly monoclonal antibodies. Just so you know, there's a couple of housekeeping items here today for the people that are participating. You'll be able to ask questions by a chat box. For the people in the audience, we're going to reserve questions till the end if you have any, and then we can go ahead with the survey questions. We want to be able to give feedback on what we've been giving you, and then of course there's a course evaluation. So with that in mind, let me give you kind of an overview of what we're going to talk about at a high level. And from the things that I'm going to speak about, I could probably spend a week on each one of these subjects. We don't have time for that. I'm going to talk about things at a macro level. The main thing is I'm going to tell you about what is probably the fastest growing source of new medicines. This is monoclonal antibodies for therapeutics. I'm going to review the approved mAbs and the target classes that have been out there, a little bit about mechanism of action. I'm going to talk about how therapeutic antibodies are actually generated, some of the technologies that are being used and in play. After that, a little bit about the pharmacology. This is a pharmacology class. Large molecules are different than small molecules, so I'm going to get into a little bit about that. And then tomorrow, we're going to talk more about the practical aspects, how you merge the science, the intellectual property, and the business to actually drive value for these going forward. And this is an important aspect to understand about therapeutic antibodies that are being developed in the biotech arena, but actually also at the universities too that are marching into large pharma. So with that in mind, let me just tell you a little bit about therapeutic antibodies. So this is a list of the approved therapeutic antibodies. There's been a few additions to that. This is too small to read. There's about 36 of them that are now approved. If you consider antibody-like molecules, these are Fc fusions, peptibodies, et cetera. This list gets to about 42 of these right now. And this is a growing list. Right now, there's about 350 antibody and antibody-like molecules that are in some stages of preclinical and clinical development waiting for approval. And as I said, this list is going to get much longer. This is to tell you what the impact of that. And if you look carefully at this list, again, it's very small. This is the top 20 antibodies that are going forward in a clock of what happened in 2013 and the prediction from 2020. And one of the surprising things is in 2013, therapeutic antibodies, the top 20 accounted for almost $60 billion in revenue. Okay, it's an amazing thing. If you look in 2020, this is predicted to grow to about $120 billion. And that's an amazing feat when you really sit back and think about it. And this is why this class is growing. There are properties to there in terms of safety and efficacy, which are very desirable for the clinician and the patient, and that's why this is growing. I don't know if this will be supported by medical economics in the future, but this is where we're going. So this is an important part of your understanding of new medicines going forward, as opposed to just the small molecule market. So let's talk a little bit about the target classes. So if you think about antibodies, we're talking about things that are on the surface of the cell or outside of the cell in the circulation or in some body tissues. And if you look at that and you look at the genome, which has been completely sequenced, there's about 30,000 proteins, about 1/10 or 3,000 of those actually make it to the cell surface or outside. So these are the target universe you can think about. So when I talk about those approved antibodies, and then all of the antibodies in development, about 350, there's actually only about 150 antibodies targets that are being actually prosecuted. So there's some redundancy here. And if you look at the different classes, you can think about it, for example, cytokines. You can imagine those play a role in disease, and neutralization of those are very important. There are things, for example, TNF inhibitors, IL-6 inhibitors, various cytokines involved in inflammation. There's cytokine receptors to block those. We have immune modulators and cell determinants being involved in things like immuno-oncology. growth factor receptors, cell adhesion molecules. And then there's things like GPCRs, which are a very druggable class with respect to small molecules, but with respect to large molecules, this has been a very difficult class to prosecute, but very important. It's that transmembrane domain and making antigen that's made this very difficult to deliver antibodies. Other things include extracellular matrix proteins, mucines, amyloid, things in the complement pathway, which are being prosecuted, Ig and Ig receptors, for example, enzymes, serum proteins, things like gated voltage channels, transmembrane proteins like that, another important class, but also very difficult to prosecute because of the nature of the antigen and its intercalation within the membrane. And then we have, of course, infectious disease targets. You can imagine antibodies would be very important for that. What the surprising thing is that only a small fraction of the druggable genome with respect to antibodies are actually being prosecuted right now. And there's a whole class of novel targets out there where the biology is being brought to those to make them validated in terms of antibody drug targets. So with that in mind, let me just tell you a little bit about some of the mechanism of action of antibodies. Neutralization of growth factors and cytokines, that's an obvious thing. Antibodies that block its interaction and be able to interact with the receptor and cause signal transduction that results in some disease process. There's blocking receptors and adhesion molecules from doing their duty. There's cell killing and clearing functions, binding to cell targets to actually kill that cell, for example, in oncology, or clear cells, for example, in immune diseases like autoimmunity and inflammation. And of course, there's interference of pathogen entry and pathology, and interference of enzyme activity. This is what the world looks like in terms of the large molecules. So what are therapeutic antibodies? And I'm showing you here a drawing of an immunoglobulin, and I think people are much aware of what they look like, but they're basically composed of really two areas. There's a constant region that has many effector functions associated with that actually acts as the scaffold for which these antibodies can bind to something. And then there's the variable domain. Variable domain is composed of two areas. There's the framework regions, which I look at as kind of like the palms of your hand, and then there's the CDRs that actually touch the antigen, and these would be the fingers. The CDRs are hypervariable. They differ. They undergo very different changes to be able to mold and bind to specific targets with very high degree of specificity. There's really four classes of immunoglobulins with respect to the constant region: IgG1, IgG2, IgG3, and IgG4. Only IgG1, 2, and 4 have actually been used as therapeutic modalities, and there's reasons for that in terms of manufacturing considerations. Chiefly, IgG1 is the main class that's being used, and it's preferred because of its ability to be expressed at a very high level in a recombinant system in mammalian cells, and the fact that it has a very long circulating half-time. We'll talk about that more later. The heavy chain is bound together by cysteine bonds as a dimer, and the light chains go on top of that to form a heterodimer. The constant regions include an Fc domain, which can bind to Fc receptors that are involved in recycling and immune function, and also can bring other effector cells to, for example, like T cells and other things, complement binding, that are very important for their role in treating infectious disease, or in fighting infectious diseases in the body, but those can be also very important as the therapeutic. The hypervariable regions in the CDRs are involved in antigen binding, and there's an incredible repertoire that's present here. Up to 1 billion different types of binding proteins can be generated from the genome during the immune response. And this is based on two different processes. I'll talk a little bit about that. There's a recombination event to bring many different variable domains together, and then there's a somatic hypermutation event, which is actually a change in those to mold it to a particular antigen. And it's important to understand that biology in terms of how you'd use these to engineer to make therapeutic proteins to prosecute specific targets. So what type of antibodies are being used in the clinic, and this is just a little diagram showing you the various modalities that are being used. And I've circled the one which is the most common. This is the humanized or fully human IgG1 molecule. This is the preferred modality. Most of them look like that. But you see all these iterations of special types of molecules which are small hyphenated fragments, for example, the FABs and FAB primes. There's little mini bodies. There's mini bodies that are put together that just have the variable domain. And some of these actually have very important functions for specific disease targets, tissue penetration, et cetera, binding more than one target together, et cetera. And for example, just last week, a new product was actually approved, and it's right here, a bispecific or BiTE that brings actually T cells together by binding to CD3 along with a cancer target to cause actually immune killing of a particular cell. So all of these are being used. They are not just of human form. There's actually antibodies, for example, from the shark, from camelids, which are being a very important modality. All of these are being employed, but I'm going to talk chiefly about this here. This is the humanized or fully human IgG1 because this is the majority of things that are actually in the clinic. So let's talk a little bit now about generation of therapeutic antibodies and some of the technologies that are being used. First of all, how do antibodies get produced? And this is a little diagram just showing you what goes on during the immune response where you have pre-B cells that basically undergo maturation. They come into contact with an antigen, and there's a recombination event that brings different parts of the immunoglobulin locus together to create a novel antibody. And then there's an affinity maturation process and selection that goes on iteratively in the body to actually mature and get specificity to a particular antigen. And this is shown below. So the final molecule is actually a composite. And like I say, there's an incredible repertoire here and ability to engineer things. So if you take this biology out and put it in the laboratory and actually put it to work, you can actually create an antibody to any molecule. You can go to the laboratory and synthesize a new chemical that's never existed, and you can make an antibody to it, okay? So there's an incredible plasticity and ability to actually create antibodies both in vivo during the immune response and also in the laboratory for purposes of making new antibodies either as reagents or therapeutics. So again, just a little to reiterate, the recombination and somatic hypermutation events, these are the key things that go on in vivo. These are being employed in the laboratory to create very extensive libraries by recombination and the gymnastics you can do with recombinant DNA. And you can actually do affinity maturation, more or less mimicking somatic hypermutation to actually create molecules and actually improve affinity. And that's going to be important when we talk about the pharmacology of the antibodies in a minute. So antibody diversity and generation, recent understanding understanding somatic hypermutation, it's very interesting what goes on here. During the immunoglobulin maturation process, there's a molecule called activation-induced cytosine deaminase. This is actually a mutator protein that can go in and actually change residues specifically within the CDR sequences during this process. And this is a form of replication mutagenesis, and it occurs every day during the immune response and actually has been harnessed in the laboratory to try to recapitulate a very natural form of evolution. Some of the mutations that occur that cause binding to a particular antigen, they're not obvious. It's not easy to predict what those are, so this can be done and performed in a way that's empirical by doing kind of cyclic or iterative saturation mutagenesis and directed evolution. So in terms of generating therapeutic antibodies, how is this done for development either as reagents or for new medicines? And one of the most common ways to generate antibodies is actually to take a mouse, inject it with an immunogen, make hybridomas, clone out those genes, and then actually take this mouse-like protein and take the CDR segments out and graft them into a human immunoglobulin backbone. This is called CDR grafting. There's been some other clever ways to do that. People have actually generated mice where they've removed the mouse immunoglobulin locus, replaced that with the human immunoglobulin locus, such that when you immunize those mice and make hybridomas, at the end of the day, you get antibodies which are called fully human. There's other ways to generate antibody. People have taken out and made libraries, put them into phage, and done panning and phage display of very high repertoire libraries, up to 10 to the 11th diversity, to try to get novel binding proteins out that bind to an antigen, and then those are then grafted back into the human immunoglobulin gene product to create a fully human antibody. There's yeast display. People have made libraries in yeast where the ability to use yeast genetics to grow them, do selection by membrane display has been very, very powerful. And of course, there's other routes to humanization, and I've listed a few of the companies that are very prominent in there. And shown on the right-hand side are more or less a humanized antibody, where you can see you have the little red regions. These are actually grafted mouse sequences that have been put back into a human immunoglobulin locus or the fully human antibody. And these are the main modalities that are being used in the clinic today. Technologies that are being used. This is a big slide. I'm not going to go into it in detail, but I want to tell you that there are a number of new startup companies that are continuing to develop very novel technologies out there. They're largely based into 3 different categories. I'll call them library-based selection, where people have made libraries and done some type of selection. There's the in vivo immunization of mice, of rats, of rabbits, even of camels, et cetera, to try to get antibody-like binding domains, and they're novelty around that. And then there's cell-based technologies where people have actually used mammalian cells, phage, yeast, et cetera, to take libraries and be able to select them. And there's another important aspect of this, that regardless of the technology being used, there's limitations to each one of these technologies. They can't cover all of the antigens that are being prosecuted. No one's clear why this is, and there's advantages to some technologies for some antigens, and there's an advantage to other technologies for other types of antigens, okay? So when you think about, I have a novel target, I've made this discovery, you can't just think I'm going to take one technology and do that, because oftentimes you will not come up with something that will be an effective therapeutic. And all the advantages together have led to the utilization of all these different technologies. And a number of these companies, I've put those in red, have actually been bought or acquired by large pharmaceutical companies because they feel they're very important to have within their own house. And that's left a void, so there's always new companies coming on. So this list is continually changing over time. So what does the industry need? Okay, so I've just listed here a couple of things about mouse immunization. This is a typical thing. You immunize a mouse, you'll get 10, 100. If you immunize a lot of mice, you'll get up to 1,000 different hybridomas that you can put through some type of screening process with your antigen to get things that not only bind, but affect the biological activity that you'd wish to have in vivo. And then there's what I call the static approaches. These are the libraries, et cetera, phage, mammalian cells, and yeast, where you have a very large repertoire, you pan those, and you can get a very large library and come up with a few hits to that you can go ahead and characterize. So again, there's limitations to each one of these. That's why you continually see different technologies being developed, even as we speak today. So how does the industry, how does large pharma deal with this? And I've given you an example here where what they do is they try to mitigate their risk by acquiring a number of different type of technologies and making sure they have access to them and they can generate molecules in a proprietary way that they can take into preclinical and clinical development. One example of this is AstraZeneca, a very large pharmaceutical company. So they have a dedicated team basically to make therapeutic antibodies to see if they can develop new medicines. And they have purchased companies, for example, like Cambridge Antibody Technologies. This is a phage technology group that came out of the MRC in Cambridge. In addition to that, they've acquired the Dyax phage libraries. They've purchased more or less the Amgen Xenomouse, for example. They're working with Regeneron with another type of humanized mouse, the Xencor XmAb technology, and they actually purchased a company, MedImmune, which is a very specialized company that delivers that. So they are making a lot of bets in this area. They're saying There isn't one technology I'm going to be using. I need to have access to all the technologies so I can make sure I can develop antibodies to the targets that are interest us to create new medicine. So industry adopts redundant resources and time-intensive approaches to address technical limitations of all the platforms together to make sure they're covering all the gaps to be able to deliver therapeutic antibodies. So with that snapshot, let me just go back a step and say what technologies are actually making it into the clinic. And I've taken things and looked at it from approved antibodies, antibodies in phase 3, phase 2, and phase 1. And this is a slide that probably reflects what was going on in about 2009. But what you can see, by and large, the majority of antibodies have come by taking a simple laboratory mouse, immunizing it, taking the spleens out or the lymphocytes from the lymph nodes, making fusions and developing hybridomas as mouse monoclonal, and then going through the kind of recombinant DNA engineering to humanize this. This is still the largest source of therapeutic antibodies that are going into development. However, you can see the growth of the transgenic animals. These are the fully human antibodies where I take an animal and I inject it using the same standard hybridoma technology that's been around for a few decades, and I can get out of that a fully human antibody that I can take into it without having doing this engineering. That can speed up the timeline development and have something that's more well-behaved. And then if you look at phage, even though phage has been incredible in source of building very high repertoire libraries and the ability to do panning and get specialized binding to specific epitopes, these have not fared well in terms of the actual development of either novel therapeutics or things that are going into the clinic. And the reasons are not obvious, and I'll go into those more when I talk tomorrow, but it has to do with the fact that the binding in that environment doesn't translate to the fully human bivalent globular immunoglobulins. Sometimes they just don't work, they aggregate, or they have other properties that are not amenable to manufacturing. So there's only a few. Right now, I think there's only actually three antibodies that have come from this source that's within that approved list. So that's not a mainstream technology that's been used for therapeutic antibodies, but it's been very useful to derive reagents and binding proteins that are used for the laboratory and all kinds of studies and validation work that's ongoing. So I want to talk a little bit about humanization because it's such a major importance for deriving therapeutic antibodies. So I want you to know a little bit about it. And there's really kind of two approaches. Part one is just the standard procedure. This is a hybridoma technology. Inject a mouse, take out lymphocytes, fuse them with a myeloma cell to create a hybridoma. This is a hybrid between two cells that can grow clonally. And then by doing that and plating them, you can actually do screening and ELISA plates, get binders to the protein of interest. You can take that hybridoma out, grow it, make antibody in tissue culture, test that, validate it, and then go on to actually clone that and sequence that. And that's the actual starting point for the development of a therapeutic antibody. And this process takes probably about 6 months to do. You can speed it up, it can take longer. You can do different types of immunization routes, whether it's basically subcutaneously or in foot pads, and do all these kind of interesting things in the library to try to create different diversity. But this is the typical thing. In the average kind of campaign for a hybridoma, you can generate tens, maybe up to 100 different types of binders. If you're in the industry, you want to develop probably 1,000 different types of binders so you can screen everything to make sure you get the best protein that comes out of that. And the type of affinities that you usually see from this range in the kind of the 1 to 10 nanomolar down to the low picomolar level. And what happens in this, you get the natural immune response, you get the actual recombination, somatic hypermutation to get very high binders. The next part is actually to humanize, is actually take this antibody and then generate more or less humanized antibody from that. So you can see on the very left, this is what you get out of that process. Mouse sequences in red. The first step, you actually make a chimeric antibody. I take the variable domain and I splice it onto a human constant region, both heavy and light chain, and I get what's called a chimeric antibody. And then you can take that and then start to replace the frameworks with the human framework regions that are from the immunoglobulin locus of humans that are most closely related to the mouse sequences, and you can see you have left just a little bit of resident that. And then, of course, if I use a xenogenic mouse or a transgenic mouse, I can come up with a fully human antibody. And at the bottom, and this is very important, when you put these into a human, you can see more or less the immune response to these, and it's important because as you can understand, things that have part mouse sequences in them can be very immunogenic. For example, if you have 100% mouse protein, about 60% of the time you'll get immune response, which actually neutralizes that. And there's only one approved fully human mouse antibody, and that's used for only singles ejections, and that's been used to target CD3 during more or less creating during transplant rejection. Chimeric antibodies, there's several of these that are actually on the market. One of them is a very famous protein called rituximab used to treat lymphoma. And about 30% of the time you'll get immune responses to that. So you can imagine you can't dose this continually because eventually your own immune system will actually take this out and neutralize it. And then there's humanized, where about 1 to 8% of the time you'll get immune responses to that. So this is actually tolerable and can be used over and over again. And then there's the fully human protein. And about 1% of the time you'll get immune responses to that, we reject that. So this is, you can see, going from old technology to the newest technology, what the driver is to get things that are very low immunogenicity. So they can be used over and over again and be tolerated in the immune system. One perfect example of dehumanization, this is one of the first humanized antibodies that have been approved. This is a molecule called Herceptin or trastuzumab. It binds to a receptor called the HER2 new protein, which is present on breast cancer cells and a very important treatment for a subsection of breast cancers which have an amplification in the HER2 gene. And this is a little bit what's going on at the kind of molecular level during the humanization. And what happens is you take the mouse sequence and you define what the CDRs, and this can be computationally done and by structure function analysis. You basically remove those and you splice that into a human immunoglobulin gene variable region, which is most closely related to that mouse sequence, and put that back in. But oftentimes when you do that, you lose the original affinity. So the original mouse antibody that used to generate Herceptin had an affinity of around 3 nanomolar. And then when you did actually the humanization without any back mutations, basically you lost about 10-fold in affinity of about 25 nanomolar. And this is not useful for the clinic, and I'll talk about that in a while. So what the scientists did at Genentech, they went back and added back a few of the mouse sequences. These are called back mutations. And what they did, they reduced the affinity to around 250 picomolar, or 3.3 nanomolar actually. And this was very useful for the clinic. And because of that, there's some issues with immunogenicity, but these are very well tolerated, and this is very important medicine that's been on the market since the mid-90s, and it has a very important effect for patients with breast cancer. So this is a typical example of how things have been done in the past. This is kind of the plain vanilla fudge way to go ahead and make a humanized antibody. There's other processes you can go, aside from what I call the wet approach. You can actually just start with a sequence, a known sequence of an antibody. Get a hybridoma or look at a published paper or look at a patent and just directly sequence it, and I can do CDR grafting and directly do that. So I don't even have to do the immunization. And today, when we look at how many monoclonal antibodies are out there in the public domain, you can actually just go in and take those sequences, and it's called the dry approach, and directly engineer those into a humanized form of an antibody, and actually create a new molecule, new intellectual property, and new methods of use using that approaches and the typical kind of recombinant DNA gymnastics that you can do in the laboratory. I wanted to show you one other technology, and this is how things have been more or less done. This is a process that I was using in collaboration with VivaMab and BioAtla. I call this framework grafting and not CDR grafting. So instead of taking CDRs and putting in one immunoglobulin locus, you actually take a library of all framework regions that are used in proteins, antibodies that have been in the circulation, and you graft those randomly next to the CDRs, and then you do screening for those, and you can make a large library to find out what is the best match between the frameworks and the CDRs that you want to use. And you do this by putting the library in mammalian cells, so it's well tolerated. In fact, in the same expression host that you're going to manufacture in. And you can select out antibodies from that that don't have this reduction in affinity and can actually improve affinity. And to kind of demonstrate that, I'm showing you actually a ELISA test of a number of clones that came out of a process with that. For example, these are 33 hits with a specific activity that's greater than or equal to 75% of the wild type activity. And that bar across shows you what the wild type activity are. And you can see some of these actually have improved affinity above that. So in one-step process, I can create a humanized antibody and actually improve its function. This is a very important aspect. At the same time, you don't have to go back and make other iterative changes in the CDRs and frameworks to improve affinity to meet your design goals to treat a particular disease. So immunogenicity, is it an issue in the clinic? I've shown you some data here with some basically marketed drugs and the immunogenicity profile for those. And you can see the chart there on the left-hand side. Basically, if I look at this, you can see there's the immunogenicity from marine proteins, chimerics, humanized and human, fully human antibodies. And by and large, immunogenicity has not been an issue for monoclonal antibodies, okay? What happens is sometimes people basically get an immune response, particularly an antibody, well, you can switch to another particular antibody and try it instead. Since there's this redundancy in antibodies to specific targets, a clinician has the ability to take one product, use it for a while. If there's an immune response, they'll switch on to another product, et cetera. There's really been no pathology associated with immune response, but there is this drive to become as human as possible, both from the terms of the therapeutic efficacy, but also from the marketing standpoint, so people can do that. And one classic example here I'll show you, I'll talk about in a second, is the antibody Humira, which actually is the largest selling medicine right now that's on the market, and then Remicade. Remicade is a chimeric antibody. People get immune responses to it about maybe around 30 or 35% of the time, so it's going to be used for a while, and then they switch on to Humira. Humira, even though it's a human antibody that came from phage library, they had to do genetic engineering to that altered its human structure, so about 16% of the time people get immune responses to that. But it's well tolerated and people can dose around it, but it's not created any it's not created really any problems in the clinic, but there is this drive. So in general, immunogenicity of therapeutic antibodies has not been a major issue in the clinic. If you look at the various technologies that are being used and the timelines to really get them to the point where they're ready for clinical study, filing an IND, you know, I look at this and some are a few months short and they all take about the same amount of time. So if you came into the laboratory today and say, I've got a great new target, I want to drive an antibody, it will probably take you about 12 to 18 months before you've gotten this ready to basically send it to manufacturing and file an IND. Okay, that's about the time point. You can shave a few months off that. Sometimes it can take longer, but you're talking about 18 months from the drug concept, the idea, starting an immunization campaign, and getting an antibody that's ready for manufacturing and doing the critical pre-IND enabling studies to actually take it into the clinic, regardless of that. So, you know, it's interesting to think about the technologies you use. You can shave a few months off, but people actually use a number of these technologies in parallel to try to be able to get to a point where they have a drug candidate that can go into clinical development. What can you do to enhance efficacy? Okay, there's a number of technologies out there to try to spice up the effects that are out there. And there's a variety of things, for example, to change the glycosylation state. People have found that actually the glycosyl residues, if you remove the fucose level, you can massively enhance the antibody-dependent cytotoxicity associated with that, or ADCC, by bringing effector function into that. And that's been a very important part of glycoengineering for drugs that are used to targets for the immunoglobulin range. For the FC engineering, you can also enhance ADCC, et cetera. And there's also other technologies, for example, using antibody-drug conjugation by actually taking the antibody, using chemistry to tag on a cytotoxin to that to create a therapeutic cytotoxin. There's a number of technologies out there that are used in that. So you can actually do things also with the FC engineering to enhance F-life by binding to the neonate FC receptor, so things are cycled, so they can have very long half-lives and presence. So there's a number of technologies out there that are being applied to actually enhance the activity in the clinic. About antibody-drug conjugates, this could be a whole week seminar to talk about the technologies and that are being used to that. There's a variety of different cytotoxins that are being directly coupled to the antibody to create something that can actually bind to a cell, get internalized, and release a very potent cytotoxin. So cytotoxins that normally are too toxic to be applied as both small molecule drugs now can be attached to an antibody, targeted to a particular cell type, and create cell killing. There's now 2 approved antibody-drug conjugates, Adcetris and Kadovia, and these are paving the way to a whole generation of new antibody-drug conjugates, and there's been a lot going on to create very potent cytotoxins that can target, for example, the microtubule apparatus involved in cell division, and also, for example, targeting the DNA in a variety of ways, binding to DNA, stopping replication, et cetera. These are kind of major pathways that are being going on. So this is an area to really watch and understand about that. And the whole creation of these, the clinical development of that, the conjugation, the linkers that have been used, the trigger for release is a whole area that's incredibly exciting, that's merging the biology and the chemistry together. So I want to talk just a little bit for a second about the pharmacology. This is the pharmacology course. I know you guys probably know and understand a lot about small molecule pharmacology and the delivery, half-time, metabolize, et cetera, excretion, all of these things. Antibodies, it's a little bit different. By and large, an antibody kind of comes with a certain pharmacology of that. If you look at the half-life antibodies, whether it's targeting this antigen or that one, they're about the same. They have a half-life in human, so around 21 days if you're an IgG1. If you're an IgG2, about the same. IgG4s have a much shorter half-life, and they all get used. But by and large, that kind of comes with a molecule per se. There's a few concepts that are really important to understand the biodistribution, the pharmacology of antibodies. For example, the actual specificity for the target really has a very important control of pharmacology and biodistribution. And the efficacy safety are generally correlated with the extent of the interaction with the target antigen. When you talk about affinity constants, it's really the off rate, which is the important driver for biology in vivo. Achieving the appropriate affinity. There's a lot going on in understanding the density of the target and understanding the actual optimized affinity, so you can go back and think about that. And I just wanted to give you an example. I talked about Humira and Remicade, for example, and that when you look at the affinity, so I brought 2 little things. People here may not be able to see this on the screen, but this is basically a dose of Remicade, okay, about 500 mLs. This is not actually Remicade. I didn't have a bag of it. This is a dose of Humira. It's about 800 microliters in a tuberculin syringe, okay. This, you go into the clinic, okay. You drive there, you sit there, and you take your blood pressure, you go in there and you get an infusion. It takes you about an hour. You sit for a little bit longer. They give you some Benadryl to make sure that. It takes about a half a day, and you get one of these twice a month. Okay, this comes to your home by Federal Express. You put it in the refrigerator. It has about 800 microliters. You basically give a subcutaneous injection. You can be watching TV and eating popcorn at the same time, and that's your dosing. So this was a first-generation TNF inhibitor, okay? Very important drug for people that have, for example, rheumatoid arthritis, Crohn's disease, psoriasis. This is the third generation. So the difference between these two all came from antibody engineering, understanding the patient, and understanding what was actually a breakthrough in this. And this is now the leading medicine of all medicines, not just large molecules, but small molecules, et cetera. So these concepts about pharmacology are extremely important to understand. Okay, binding affinity and design considerations, getting back to the Remicade and the Humira story and things like that. It's very important to understand target concentration. Is it a free molecule? Is it on the cell surface? Is it in the circulation? Is it buried in tumors? The fraction of antigen that's bound or tied up in some other biology, antigen turnover rate, the antibody pharmacokinetics and biodistribution properties are very important. Do you need recruitment of any effector function? Do I need to just bind something, or do I need to bind something and bring in immune effector cells to kill a cell? And then there's the impact of administration route, okay? Very important. For some indications, this is key. If you're a cancer patient in the hospital and you're hooked up to IVs, probably not really a difference there. It's just a matter of efficacy, not this. So there's all these design goals and consideration to take into consideration the patient, the disease, and the compliance that are associated with that. So slide number 1, impact of target concentration and dosing. So you can see here this graph here where you look at affinity going from very low affinity to high affinity, left to right, and then the actual dosing. So if you're in an area where the antigen is much less than the actual dissociation constant, the off rate, affinity is very important. You can see the impact on the dose you have to give versus the affinity. So in this particular area, driving affinity down lower is very important because it determines the actual amount of antibody you want to give. And I wanted to show you just an impact of that alone in this. This is actually some studies I was involved in where a number of different molecules were generated to the same target. These are all rank ligand binding proteins that are involved in treatment of pathological bone loss, and you can see generations of those molecules going from recombinant Fc fusion proteins, et cetera, in very different iterations, and finally to an antibody at the end. So one thing I'd like to point out is all of these had about the same affinity for the particular target, all right? What was different in all of these, if you really look at the half-life and the potency, it was all driven by basically the pharmacology of the antibody. The antibody had a very long half-life. Some of these had a very short half-life. If I go from the very left-hand side to the right-hand side, the dosing was such of a protein that was given about once every 2 weeks, and the antibody is given about once every 6 months. So you can see the impact of that. Again, the final antibody format is being delivered in a tuberculin syringe, a very small dosage, once every 6 months. The antibody on the right, on the left-hand side, that's given in a very large volume about once every 2 months. So you can understand the importance to understand the pharmacology and the actual target density. So how much does really improving affinity do for you? And this is by changing the CDR sequences like that. If you're in a position where there's antigen concentrations is very high, particularly if it's on a cell surface, actually driving affinity gets you to a certain point where it doesn't matter anymore. So going from, let's say, a 50 picomolar antibody or 100 picomolar, driving it down to 1 picomolar really doesn't change the dosing. So you have to understand really the target biology and concentration to understand what your design goals are to create a therapeutic antibody. Here's some pharmacology again. I'm showing you 2 different types of rank ligand binding proteins. AMG 162, this is a therapeutic antibody. It's now marketed as denosumab, and versus a Fc fusion protein. This is done by single dose kinetics in cynomolgus or non-human primates. And the red on the left-hand graph is a therapeutic antibody, the Fc fusion protein. You can look at the difference in the pharmacology of these 2 proteins, and it's really driving. Again, both of these have about the same affinity. And if I look at actually the pharmacodynamics of that, on the right-hand side, you can see actually the decrease in bone resorption markers, and you can see the impact of an antibody versus the Fc fusion protein. So again, as you're designing these things, understanding affinity, understanding the pharmacology really changes the actual delivery to the clinic, and it really makes the decision about what you really need. Sometimes short half-life is what you want. I don't want it around for 6 months. Okay, how do I withdraw or take a patient off that? Sometimes you want it because it's very convenient to give something that lasts a very long time and the patient doesn't have to come into the clinic over and over again. So understanding these aspects are very important. So as we kind of wind down here, I just want to give you kind of the takeaway points that I've talked about today. So therapeutic antibodies, this is really a very large growing class. They're safe, they have great specificity. Hello? Yes. Okay, there we go. Again, the takeaway points, antibodies are generally very safe. They're very specific, okay? They don't have a lot of off-target things. And we're going to talk tomorrow about the development of these. Really, you know, once you get efficacy in the preclinical stages, they go very well through the phase 1 safety studies because they're generally not having off-target side effects, et cetera, that have to do with metabolism or hitting other pathways. There's a very diverse targets that can be prosecuted by antigens with prevalence for specific classes. We've talked about those. I think the class is going to grow as we understand more novel targets and associate those with disease pathways. Some of the things that are very easy and no-brainers are blocking cytokines or hitting receptors, creating cell killing. There's whole classes of proteins out there like G-coupled proteins, ion channels, et cetera, I think which are going to be very important classes of new medicines, but they've been difficult to get antibodies to, but actually using newer technologies, people are going after that in the future. There's multiple modality options. There are all kind of flavors of that. We can use full-length, bivalent, humanized or human antibodies. There can be antibody fragments. There can be fragments tethered together to make bispecific things hit more than 2 targets at once or 3 targets at once we want. There's antibody drug conjugates where we're actually taking potent cytotoxin and putting them together. PK-PD relationships is very important to understand design goals. And usually when people develop therapeutic antibodies, they're thinking first about the clinic. What is the unmet need? What do I want to get at to? It may not just be a new target, new antigen, but it's actually proving the antibody for better patient compliance and delivery. For example, infusion versus a subcutaneous injection. And there's other properties that we're going to talk about more tomorrow when we get into how you manufacture these. These are complex, large molecules, okay? Being able to manufacture them at a high level in a way that they're very reproducibly and have the same identity is very important. And integrating this manufacturability, being able to generate the IP and the science and the target biology and the design concept is very important aspect to making new antibodies for the future. So with that in mind, I think we've talked for about 50 minutes. I want to open up this to discussion both with people here in the audience and also for people that are at the webinar. I'm happy to take questions. I believe if we're going to take questions here, we would like you to talk on the microphone for people on the webinar can hear. And then I think we'll be able to take questions basically from people that type into the webinar. So with that in mind, that's a general introduction of therapeutic antibodies. We're going to go into it much more detail tomorrow where we talk in depth more about the technologies, the intellectual properties that you need, the manufacturing process, and integrate that together with the science to how to actually produce an antibody, and then to go on and actually create something that can go from the laboratory into the clinic. And a lot of what this is getting at is to understand that the early steps of generating an antibody are not so difficult. They can be done in small companies, it can be done at the university, but you can create a lot of value along the way by doing things in a very specific manner to come up with something that's very potent and novel that could be partnered and delivered to a large pharmaceutical company through licensing or partnering interactions. So with that in mind, thank you very much for your attention.